Just when you think day one couldn't be any busier, there are still things to do, such as beginning the secondary treatment process, cleaning up from day one, and of course, set up for day two. Let's start with secondary treatment.
21, was that a change? Yes, that was a pretty big change. So without ever using any energy, we were able to decrease the suspended solids in our wastewater just by letting it sit. Okay? While you guys were doing the lab, your tests, Rita took off that middle layer of suspended water, which was mostly just water. Okay? The suspended stuff is left behind, the plastic, the coffee grounds, the toilet paper, and that kind of thing. And so now what we're getting ready to do is start secondary treatment, which is the biological component where we're going to put in bacteria that are going to do some work.
Secondary treatment is a biological process that uses microorganisms and oxygen to remove nitrogen waste and organics from wastewater. With your students, discuss secondary treatment and how it connects to the nitrogen cycle and eutrophication. The microorganisms will use the pollutants, ammonia, sugar, protein, and carbohydrates in the wastewater as food for themselves. These microorganisms are a key participant in the process of cleaning wastewater. It's important to recognize that microorganisms are susceptible to harsh chemicals and environmental changes such as temperature. Pouring excess household chemicals or paint down drains can disrupt the microorganism population. Take these harmful items to a hazardous waste site for proper disposal. Transfer tanks. Aeration tanks will be used to simulate the secondary treatment process, so we need to transfer the primary treated wastewater to the aeration tank. Be careful not to disturb the sedimentation tank while transferring. Double check that the valves on the aeration tank are closed. Follow the directions provided in the lab book to transfer wastewater from sedimentation tank to the aeration tank. Connect air pump to the tank and plug it in. Then open the valve connected to the air pump. You should see bubbles in the water. Now you know it is providing the microorganisms with the oxygen they need for metabolism.
Could have been any one of us in this room that contributed to the RAS sample. This is exactly what you think it is. So we are introducing microorganisms into our wastewater supply to do the work.
The treatment plant should have provided you with a microorganism sample called RAS, or return activated sludge. Wearing your goggles and gloves, add about 50 mL of the RAS to the aeration tank. Allow to aerate until the next time your class meets. Helpful hint, too much RAS is not a good thing here.